Thank you, Anna, and welcome, Kyle. It's great that we have you joining us virtually today. Kyle got his degree as an undergrad actually in computer science and then continued as an MD PhD student at Harvard and MIT and worked on something that was also very dear to my heart at the time, namely microRNAs. So he did his PhD part in Dave Bartel's lab and actually published some seminal papers about patterns of microRNA targeting and conservation of microRNA targets during his PhD, and actually worked also together with the lab where I did my postdoc with Chris Burge at the time at MIT. And so our paths almost crossed, I guess, or maybe even directly crossed a little bit, but I soon took my first faculty position after. And then Kyle actually continued to finish obviously his MD and then also residency in genetics, and then stayed in Boston at first for a postdoc at the Broad Institute with Daly and Bernstein, and then took up his position at Illumina, that very early on started developing their own AI department to actually not just provide the technology that probably most of us have used, the actual machines, but also to work on computational interpretation of the data. And also very early on got into deep learning models and how they can be used and applied in meaningful ways in genetics. And one of the first seminal papers from that time is actually SpliceAI, which again connects to stuff I did in my postdoc. And so we read that paper up and down when it came out about 5 years ago, to actually manage to develop and utilize cutting edge AI technology in a way that we can interpret what these models are doing, and so that they're in this way useful for genetics and in a clinical use. And I'm sure Kyle is going to tell us a lot about what he's been up to in the last few years and where the field stands at this point. Welcome, Kyle.
Thank you so much for the kind introduction. Yeah. Um, Let me see. Everything working? You guys can see my slides?
Yes, we don't have the full screen view yet, but hopefully. Um, so we saw your full PowerPoint or Keynote, I don't know what it was, window, but we don't see the full screen at this point. I think we lost Kyle there for a second. I think he joined again.
Okay. Everything working now?
Let's see. Yeah, now it's
Okay,
working.
great. Um, all right. Yeah. So I'll say a bit about um, my group at Illumina and what we work on. So uh, I think 99% of Illumina works on the sequencer and instrument and everything that goes around that. We're kind of the, I guess, 0.5% of the company that focuses on sort of basic science research and just trying to figure out how to make sense of all the data that's being generated. And I think long term, Illumina Knots is hoping to not only push the field forward in terms of sequencing hardware, but also in terms of how we can interpret this data. Um, so one of the first goals that we have focused on is how to interpret these variants of unknown significance. And we're we have different sort of small teams within a group that are all focused on sort of different aspects of this task. So I'll start off by talking about the work that we're doing on the protein coding side, and then the non-coding side, and then I'll say a little bit about the experimental work that we're doing with Perturb-seq to generate data to um, help sort of create the training data sets that can help us tackle these problems. Um, So starting with the protein coding side, this remains a enormous problem where databases like ClinVar are growing by a few thousand variants per year, but given that there's 70 million protein coding missense mutations possible in the genome, uh, this is going to take hundreds of years before we even get uh, the protein coding genome interpreted. And yeah, unfortunately, that's the part of the genome that we understand the most about, and the other 99% that's non-coding is even more challenging. Um, so to try to solve this problem, because these variants are extremely rare, it's just very difficult to do it with traditional statistical methods. We really turn to deep learning to do this. And our intuition here is that when you're trying to interpret a human genome clinically, probably the most valuable data set for you is the allele frequency tables from Nomad or before that 1,000 genomes. Because you know that once a variant is common enough in the population, it's very unlikely to cause any substantial deleterious effect because natural selection would simply have removed it. Um, so I was chatting with Daniel MacArthur and several years ago, and we're just thinking about, what would be sort of the next populations to bring into 1,000 genomes or Nomad to try to identify these common benign variants. And we kind of realized that actually, well, it's pretty difficult because the remaining human populations tend to be very isolated. They tend to not respond very well to you trying to contact them. And ultimately, we're all just bottlenecked out of Africa a relatively recent, time frame ago. So humans just don't have that diversity. So thinking about that, we first started looking at common variants from other primate sequencing cohorts. These are all very small at the time, less than 100 primates. And what we discovered was that variants which were common in other primate species tended to also be benign in human. And you can verify that by, overlapping variants that are common in primate species with variants in a ClinVar database. And you find that overwhelmingly these are benign. Um, so we took this approach out and we essentially got together a consortium of all the sort of primate geneticists around the world. And we essentially offered the sequencing to do this project. And altogether, we collected about 800 individuals across 200 species. And this allowed us to essentially do 1,000 genomes, not of different human populations, but of different primate species. So it's about like 4 individuals per species. So we're largely just collecting the common variants present in each species, and we map these back to human. This gives us a database of about 4.5 million missense VOSs that are likely to be benign based on the fact that they occur at high population frequencies in their species, and these primates have within conserved regions are more or less identical to human in their protein coding sequence. So the effect of a variant in primate is very likely to be conserved in human. Um, so this gives us a data set that's about 70 times larger than ClinVar, and now we have enough data to do some pretty good machine learning. So to give you a sense of the power of this data set, each dot here is a pathogenic variant in ClinVar, and the x-axis is the 2,500 amino acids that make up the length of CACNA1A. This is a human rare disease gene for epilepsy. And yeah, so red is the ClinVar variants that are pathogenic. Blue X's are the benign variants we got out from primate sequencing. So you can see that they are really nicely anti-correlated. And up top, you can see the same data from Nomad. It's actually the same pattern as the primate variants, but just much, much noisier. And that's the power of looking at common variants across 200 species versus just sequencing our own species excessively and just getting out all of these rare variants. So that was a case where benign and pathogenic variants separate really nicely in linear space, but in many cases they don't separate that cleanly linearly, but even then they separate really nicely in 3D space. So on the left here, this is STK11, colon cancer tumor suppressor. So on the left, I've superimposed in blue the common primate variants, and then on the right, the ClinVar known pathogenic variants. So you can see that they're in completely different parts of the protein in the 3D structure. And yeah, we can actually now take these 3D structures from AlphaFold 2, voxelize them, and then superimpose upon them all the benign variants. So then from there, we're able to infer the pathogenicity of each variant. Basically, the loss function is we try to predict the probability that that residue or that variant will exist as a common primate variant, meaning that it will be tolerated by evolution. So we never actually need to train on any human clinical data at all. We can just basically infer pathogenicity based on the absence of primate variants within those regions. Um, I'll note that this approach is very good for helping us find which residues are going to be the most pathogenic. But then among the residues, there's going to be certain substitutions that are more or less pathogenic. And for that, you actually need protein language models to really get, fine-scaled resolution. So we actually bring those also into the loss function. So the combination of those two really helps you narrow in on what are the most pathogenic residues, and then among those pathogenic residues, what are the tolerated substitutions or not. So you can see over here, this is the performance of PromoterAI 3D in orange. The other methods are in gray. And yeah, just starting from the upper left, this is performance on 9 different genes, correlation with saturation mutagenesis assays. So functional, in vitro assays in cells where people made all mutations. The middle one is a correlation with clinical quantitative phenotypes in UK Biobank, 500,000 people. So this is looking at correlation of your predictions for people who are rare variant carriers and who carry rare variants in genes which have clear effects on quantitative phenotypes like glucose or cholesterol levels. The upper right is AUC, separating benign and pathogenic variants in ClinVar. And in the bottom is 3 large rare disease cohorts: DDD, intellectual disability, ASD, autism, and CHD, congenital heart disease. So in each of these cases on the bottom, we're looking at separation of missense mutations that occurred de novo in patients versus missense mutations that occurred de novo in healthy sibling controls. So you'll note that we did very well on these, but also that there was no other method that was number 2 in more than one comparison. So if you actually take the average, it's quite a large gap between us and the other academic methods. And then, yeah, a few months after we published this, DeepMind also published Alpha Missense. Alpha Missense is actually quite good. It's has many similarities to PromoterAI 3D. For instance, it even trains on primate variants. But that's no problem. That's how science advances. They borrowed many things from us, and we borrowed many things from them, and that's how it goes throughout the community. So because of this, there was no actual direct benchmarking of the two algorithms since they came out so close together. So we've actually released a preprint where we now show that you can actually use the UK Biobank proteomics data as a really large, real-world cohort to benchmark different interpretation algorithms. And the way it works, as you can see on the right, so for UK Biobank, there's 50,000 people who've had 3,000 proteins measured using Olink proteomics assay. And with these two genes on the right, both rare disease genes, you can see that there's a nice correlation between the PromoterAI score. So the more pathogenic your PromoterAI score, the lower your level of that protein. So the y-axis is the z-score of that protein level. So the most pathogenic missenses are actually pretty close to loss of function, nonsense or frameshift mutations. So this is essentially because most pathogenic missenses actually act by destabilizing the protein. And therefore reducing the protein levels. So on the left here, you can actually see that by using this cohort, which is like 3,000 proteins, 50,000 participants, you now have kind of the largest real-world data set that no one's ever trained on to benchmark various methods. So you can see that PromoterAI is indeed best. Alpha Missense is actually pretty close to us, followed by ESM from Facebook, followed by E from Debbie Marx's lab, and yeah, and then sort of all the other methods are quite a step back. So I think what is really exciting is that the deep learning methods really do seem to be quite a bit better than the previous generation methods on this data set that no one's had a chance to see before or benchmark on. Um, yeah. So I'll say that it takes forever to get these papers published nowadays. So, um, in our original paper that came out in Science 2023, that was about 800 primates, but now we're up to about 3,000 primates. So we're hoping to submit a new version of this probably near the end of this year. So it has a lot more data as well as many more improvements on the algorithm. So we'll put that out and then we'll see what DeepMind's response is. But I think one particular exciting thing about this new primate data set, which is about 4 times larger than what we've had before, is that we now have really dense maps of primate variation that are tolerated. So not only does this apply to coding regions, but also to non-coding regions. So in the non-coding regions, we estimate that if the nucleotide can accept the mutation, there's probably about a 75% chance that we'll observe it in our 3,000 primate data set. This means that you can now actually tile the non-coding genome and identify regions of negative selection. So here you can see in the LDLR promoter, three kind of small mutational deserts where we never see any primate variants, and they correspond very nicely to conserved transcription factor binding sites in the promoter. Here's another example. This is a enhancer, but you can see, yeah, again, a few of these examples of locations where you have really nice depletion of primate variants exactly over the motif. Yeah, also one example over here, STAT2, where there's a very clear motif and very clear depletion, but no conservation. Reason is that this appears to be a very recently evolved site. So I think it was very nice to have the Nomad whole genome papers come out recently. But when they tried to define regions of constraint using just human sequencing data, because so many of those are rare variants, they could essentially only find kind of 1 kb regions of constraint. Here we can really define constraint on a almost nucleotide level, which I think will be really exciting for non-code interpretation going forward. Um, All right, so the second piece I want to talk about was our second paper that accompanied our Primate AI 3D paper in Science about a year and a half ago. And this was applying Primate AI 3D to large-scale biobank cohorts, in this case, a UK biobank. So after we'd shown sort of its effectiveness in rare diseases, we applied it to common diseases. So on the left here, you can see each dot is someone in the 500K UK biobank. And then the x-axis is the PromoterAI score. The y-axis is their LDL cholesterol level on blood draw. So first of all, you can see this really nice correlation between the two, where variants who have scores of 0 have almost no effect on cholesterol levels. Those with scores of 1, most pathogenic, increase your cholesterol level by almost 2 standard deviations, which is what you observe for frame shifter nonsense mutations. Obviously, this is not a perfect correlation, it's still very noisy. The reason is that your cholesterol is dependent on many non-genetic factors like diet or exercise, but we estimate of the total genetic variance, we can explain about 2/3 of the variance using Primate AI 3D now, and we're constantly looking to improve that. And then, yeah, on the right here, you can see an example of a very interesting gene, PCSK9, where typically the more the gene's function is disrupted, the lower your cholesterol level gets. So mutations like loss of functions that break this gene are actually protective. So this is actually what many pharma are searching for now. They're looking for genes where naturally occurring loss of function mutations confer a beneficial clinical effect. Then they can phenocopy that genetic mutation with an antibody or small molecule. So then on the right over here, you can see that by applying Primate AI 3D, you can actually substantially increase the number of these potential gene phenotype, associations or candidate drug targets that you discover, because you can filter out many of the missense mutations that do nothing. So by sort of clean up your signal, you can get much better detection of these things. The second thing that you can do with PromoterAI 3D and these large biobanks is that you can apply it to do improve the genetic risk prediction. So uh, hopefully everyone's pretty familiar with how polygenic risk scores work. So typically the way you've done polygenic risk scores in the past is that you take all the GWAS index SNPs and then you sort of sum them all up for your phenotype, and that gives you the score. The trouble is that one, this works, quite well in Europeans where your GWASs were typically conducted. But when you port them to a non-European population, the performance is quite a bit worse. So you can see that over here. Each dot is one of 70 different clinical phenotypes in UK Biobank. The dotted line is the European level performance. Orange means the common variant PRS, and then blue means the rare variant PRS. So the rare variant PRS is essentially same idea, but instead of adding up the uh, common GWAS variants, we add up all of the rare variants, essentially take the PromoterAI score, and then we multiply that by a coefficient for what is the effect of that gene on your phenotype. So you can see that in comparison to the European level performance, the rare variant PRS in orange performs just about as well in non-Europeans as it does in Europeans. And then the common variant PRS in blue is quite a bit worse in non-Europeans, as you would expect. And the reason ultimately is that these GWASs being done on Europeans are kind of overfit, whereas the rare variant PRS, it's essentially just a PromoterAI score. The PromoterAI was only ever trained on primates, not on humans, so it doesn't have, any capability of overfitting on different human ancestry populations. So that's a really nice feature, and I think the other really nice feature of getting your PRS through rare variants rather than common variants is that on the left here, you can see that the effect size of these rare variants is typically about tenfold that of the GWAS index SNPs. So this means that if you're trying to predict sort of overall variance in a population, indeed, the common variants from GWAS are better at explaining a higher share of variance, since these rare variants are ultra rare, usually less than 0.1% of the population. But if you're trying to find those people at greatest risk of a disease, those are actually easier to find using the rare variant PRS because you can get there with just, one or two, variants, whereas with common variants, you kind of have to hope that all of these vote in the same direction, which is very unlikely statistically. Um, Okay. And then, yeah, just want to take this back to, drug discovery again. Ultimately, I think many pharma are really excited about this. These large-scale cohorts like UK Biobank, and then most recently Vanderbilt University, and I think all of us in the UK, sorry, not all of us, our Future Health in the UK, all of these are funded to some extent by pharma who are looking to find these new gene phenotype associations. I think ultimately our ability to discover these new PCSK9s in the future is going to be a function of cohort size, but it's also going to be a function of the quality of our variant interpretation. And to sort of just provide an example of that, we showed that if you apply PromoterAI 3D to UK Biobank and as an example, try to retrieve all of the rare variant associations for LDL cholesterol level, you find about 48 genes where rare mutations will either increase or decrease cholesterol levels. And you can see them on the right here. Among these 48, we pretty much are able to recapture the full pathway that's been worked out over the last 50 years of biochemistry. You can see it's pretty much all there. And here's the full table. So next to each gene, the effect size is, for the typical rare protein coding mutation, how much does it increase or decrease your cholesterol level by? So for PCSK9, the average effect is about minus 0.9 standard deviation. So in green are all the genes which, when mutated, lower cholesterol level, and then in red are all the ones that increase it. So you can see that, yeah, among these green list, you can find HMGCR, just the target of statins, MPC 101, target of ezetimibe, ANGPTL3, and PCSK9. So all four drugs that have been approved for management of high cholesterol, we can just rediscover right here. using a large enough cohort. I think the tricky part going forward for the field is that this is relatively easy to do for cholesterol. Um, but for any other disease, like Parkinson's, or multiple sclerosis, we consider these common diseases, but they're actually about zero point 0.1 to a couple percent in the population. This means that, unlike cholesterol, which is measured in everybody in the UK Biobank, most of these other common diseases only affect a few thousand people in the UK Biobank. So uh, yeah, once again, you're very underpowered, especially to find protective effects. So it does look like to really do this at scale and discover the sort of next drug targets of the future, it's not 500,000 that we need, but probably something more like 100 times that. But I think, yeah, that's something that's going to happen probably sooner than people can imagine. And I think it's going to be really exciting for human genetics and precision medicine. Um, okay, so I think I'm about halfway through the talk, and I've only talked about 1% of the genome, so I'll try to speed up for the other 99%. So I mentioned at the beginning that one of our efforts is indeed on protein coding side, but we're also really interested in non-coding genome. So we have teams that are working on the successor to SpliceAI. We're also working on things like promoters, enhancers, 3' UTRs, microRNAs. So really trying to interpret every non-coding variant in the whole genome. We may not get there in one step or one paper, but the hope is that over the next 5 to 10 years, we can put out papers, and together with the field, we can really make some progress in understanding these things. Um, so for Splice AI, I'll quickly run through it, but since many people are familiar. But essentially, this was a residual network that could take the sequence of the gene body and basically learn a function that mapped it to the intron-exon boundaries in the gene. So this allows you to essentially take a variant, put it in, and then SpliceAI will tell you if there's any changes to the intron-exon patterns. If there are, it calls that as a potential cryptic splice mutation. And yeah, I think another thing that I just wanted to mention that's pretty exciting is that the ClinGen consortium recently adopted SpliceAI as sort of the gold standard tool for rules for non-coding variant interpretation for clinical labs. So really exciting to see this being adopted into clinical use. Um, yeah, so again, just wanted to show that, highlight a couple points about Splice AI. I think the first thing is that it compared with the previous, motif-based methods, it was able to go from about a 18% accuracy of predicting splice junctions across the genome from sequence to about 95%. And the key way that does that is that you can see for CFTR up here, each black mark is an exon, red and green are the predictions. Um, the exons are not spaced uniformly, but rather they're, quite tightly clustered together. And the reason is that splicing is actually a highly cooperative process. And yeah, that's one of the things that we are able to show in our paper that SpliceAI is actually able to model. So these were not things that you need to tell the method. It's able to sort of figure these out, directly from sequence. Another thing in the lower left corner is that we noticed that SpliceAI was much more willing to call an exon when it overlapped a region of nucleosome accessibility, or sorry, nucleosome density. So we essentially discovered that SpliceAI had learned nucleosome positioning from sequence. Again, this is not something that we had taught it. These were things that it had sort of, automatically extracted out by seeing enough training data. So these were, really exciting things to see. And we further took SpliceAI and we validated it first in GTEx on the left. So this is 800 people with both paired whole genome sequencing and RNA-seq. So we essentially looked for examples where the person carried a rare non-coding mutation as well as an aberrant event on RNA-seq. So for these sort of paired events, you could see that SpliceAI was much better at detecting them than the previous motif-based methods. And then, yeah, then we took this to a rare disease cohort for autism. Together with Stefan Sanders, we identified 28 patients with undiagnosed autism and rare de novo mutations that were non-coding. We then drew blood from these patients and then did RNA-seq on that, and we found that in 3/4 of them, you can see the aberrant splice event, which was absent in healthy controls. So we've since replicated this in a much larger cohort, 5,000 rare disease patients at Genomics England. And instead of just being sort of neurodevelopmental, this is sort of all rare disease phenotypes. And once again, we see about a 75% validation rate. So yeah, I think altogether that's hundreds of new diagnoses being made in that cohort from SpliceAI. So we're really excited to see that. Yeah, and then we're continuing to improve on this method. And I think, yeah, hopefully when the next version comes out, we'll be able to show both increased accuracy, especially quantitative in predicting splice junctions, as well as being able to show tissue-specific splice predictions as well. But the other story I want to talk about is the work that we're now doing on PromoterAI. So this is sort of the first method that is, I think, really able to crack the code of mutations under promoters. So I think this is a problem that people have worked on for a long time, but here we can show that we can actually take PromoterAI's predictions and validate them both on the protein and RNA level. So on the left here, we take this to UK Biobank, 50,000 people with proteomics data, and we just show that promoter AI variants, when they fall into promoters of these 3,000 genes that are proteomics measured, the predicted underexpression, predicted overexpression variants indeed produce those effects on the protein level in the UK Biobank. And then on the right here, we take this to Genomics England. This is 5,000 rare disease patients. And again, we show that the promoter AI score on the x-axis correlates really, really nicely with individual gene expression in that cohort. So each dot here is expression of a particular gene containing a rare promoter variant. In someone Genomics England, these are just z-scores of expression level from RNA-seq of blood draws from these patients. So given that we could predict gene expression really well, we then asked, can we actually identify rare disease patients from GEL's rare disease cohort? So what we did was every patient at GEL comes in with a diagnosis. So they may have epilepsy or they may have cardiomyopathy. So for each of these diseases, they'll have a certain rare gene panel where GEL's clinicians will look under the genes of that panel and see, is there a mutation occurring in those genes that could explain my patient's disease? So typically these gene panels might be, 50 to 100 genes, or maybe somewhere a little bit larger. Yeah, we essentially just ask, for the panel that each patient is coming in, can we actually find a rare pathogenic loss of function promoter variant that's heterozygous in that patient compared to chance? So you can see in orange that we detected, this many cases where you indeed see a pathogenic promoter AI variant in a gene that could explain the patient's phenotype. And then by chance, essentially, we just took the VCFs at GEL and randomly assigned them to each patient. So same phenotypes, same VCFs, but totally randomized the relationship between the patient's phenotype and their variants. So you can see that we have a very nice enrichment here. And then on the right here, you can see that what's interesting is that the panels originally contained both autosomal dominant and autosomal recessive and X-linked genes. So since we're looking only at heterozygous loss of function rare mutations, you can imagine that probably only the dominant genes really matter, and that's what we see here. So if you look at all genes in the panel, our odds ratio is about 1.5 to 1 compared with chance. But when you limit it to just dominant genes, your odds ratio goes to 2. And then when you limit it to just dominant genes with known LOF function, things that are haploinsufficient, your odds ratio is almost up to 3. So really nice demonstration that what we're seeing with promoter AI sort of fits the mechanism of disease. And then on the right over here, you can see that we now attempt to now look for a rare disease and say, how much can be explained by each class of variation. And the way we do that is that for each category, we just say how many variants occur in genes that could explain that phenotype versus by chance. And by chance is just, again, that shuffling procedure I said mentioned earlier. So you can see that for synonymous, this is kind of our negative control. The number of rare variants we see in genes that explain a phenotype is exactly the same as by chance. So that's totally expected. And then for missenses with very low primary AI scores, again, it's a no signal at all. For missenses with very high primary AI scores, there's a huge enrichment. Again, for those with SpliceAI scores greater than zero point 0.2, very nice enrichment. And of course, you see very nice enrichments for central splice, nonsense, or frameshift mutations. So what we can now do is we can look at how many hits we get in the real data versus shuffled. And this difference is kind of how many diagnoses can be attributable to that disease category. So now we can see on the right that roughly 80% of the rare disease cases at GEL appear to be due to protein coding mutations versus about 20% for non-coding. And that's split, yeah, between promoters and splicing. Now, I'll just mention that I think on the non-coding side, these are probably major underestimates. For example, on PromoterAI, at least in this first version, we've kept it tissue agnostic. So it's not reporting tissue-specific promoter predictions. That's something that we're looking to do in the future, but you can imagine that a fair amount of cases might be explained by that. Other things are that this is also considering only loss of function mutations. So we also know that there's many gain of function mutations caused by promoter mutations, but what we find is that they don't actually enrich within the known genes from protein coding mutations. And that kind of makes sense because the protein coding mutations are, for the most part, all loss of function. So the gain of function ones are probably in a different set of disease genes. They have different gene phenotype relationships than the loss of function ones. So I think as we expand our knowledge of this, this fraction will go up. And then similarly, I think as we bring in mutations from enhancers, UTRs, microRNAs, yeah, the hope is that instead of having a about 30% diagnostic yield for rare disease, that could be increased very substantially. Um, Yeah, in the final part of my talk, I'll just talk about the experimental work that we're doing. So our group is about one-third human genetics, about one-third deep learning, and about one-third experimental. And on the experimental side, we have been pioneering work on Perturb-seq to try to use it to interpret variants. So you guys are probably familiar with Perturb-seq, where you use CRISPR to sort of knock out every gene in a genome, and then you put that into a cell line, and then you just measure out the effects of each knockdown based on the change in gene expression signature in single cell space. So essentially turning every single cell into its own experiment. So we do the same thing, except that instead of knocking out every gene, we synthesize a library with all mutations within a gene. And now we look at the effect of the mutations, and we can measure that out based on their single cell signature. And yeah, you can sort of use known nonsense mutations and known synonymous mutations as kind of being sort of true positives and true negatives, and you can use those to train a network and then figure out what's the signatures and then classify your missense mutations accordingly. Um, so the set up the assays that we basically had Twist synthesize a library of all mutations. We insert a random barcode on the 3' end, and then we perform amplicon sequencing to map out the relationship between each barcode and the variant that is present in that cell. And now we can just use any 3' sequencing, can be 10x, can be Fluent, sort of your favorite protocol, to measure out both the effects of the variant in a cell as well as, the identity of the variant in a cell. So here are some examples. Three genes, p53, CDKN2A from cancer, SOD1 from ALS rare disease. And you can see that we have really nice separation of variants of pathogenic predictions by PromoterAI and synonymous mutations. So they have very different profiles in their single-cell RNA-seq space. But I think what's really exciting about this assay compared with the previous generation saturation immunogenesis assays that have sort of a single growth readout or single kind of assay or axis of readout, you can get these really beautiful kind of multidimensional readouts of different mutations, depending on where the mutation occurs in a gene. So for p53, what you can see is that depending on where your mutation falls in a protein, you can activate up to six different gene modules. So you might activate, TNF alpha signaling, or you might activate interferon, or you might activate hypoxia, or you might activate, G2M checkpoint or E2F, all, different extents depending on where exactly you're affecting, the domains of the protein. And yeah, you can actually sort of see over here visualized in the lower left corner. This is a heat map of correlation in expression space. And then the upper right is the correlation in a distance in the 3D protein structure. So you can see that exactly those residues which were in direct contact in protein structure, those tended to have very nice correlations in terms of the expression readout in the single cell assay. So it basically clusters into three major domains for P53. There's a tetramerization domain, a DNA binding domain, and a transactivating domain. So I think, yeah, just to make, I think, the clinicians' lives more difficult, it's pretty clear that pathogenicity is not a matter of just, benign versus pathogenic, but actually probably within many genes, there's many different flavors of pathogenicity, especially I think for the larger genes that are more complicated. Um, and depending on exactly where your mutation falls, you'll probably get different, yeah, downstream effects by single cell assay and ultimately clinically. So, yeah, I think to paraphrase Tolstoy, probably, all benign mutations are benign the same way, but probably all, pathogenic mutations, each pathogenic mutation is probably pathogenic in its own way. Um, and Yeah, I just will close out by showing a very recent experiment that we did where we made mutations in SRSF2. So unlike p53, where the effects of mutations were very much clustered along different domains, what we observe is that for this gene, which is a key RNA splicing factor, even mutations within the same domain, in this case the RNA binding domain, result in subtly different effects. So we can actually see that different mutations, depending on where they fall in the domain, they actually generate subtly different expression profiles. And not only do they generate different profiles on expression signatures, but they actually generate different profiles on splicing signatures as well. This is 10x data that I show here, but you can do this with other 3' preps. But essentially, there is enough data from these single cell preps that you can actually see a fair amount of splicing that occurs on the 3' end of the gene. So in this case, it's quite well known that SRSF2 mutations that are driver mutations for leukemia result in incorporation of a poison exon in the EZH2 tumor suppressor. And we see that exactly both in the single cell data as well as from previously published bulk data. So you can see that this mutation indeed causes much higher splicing in of this poison exon, and it results in all sorts of aberrations genome-wide. And you can see that yeah, there's actually changes to the splice recognition motif resulted caused by different classes of mutations in SRSF2. And this is not just limited to SRSF2. You can actually see this as well in genes like p53, where there's large changes to the expression profile globally. Again, not saying that p53 is a splicing factor, but probably through its indirect effects, it can change splicing recognition genome-wide. So I think, yeah, up to now, people have largely just focused on expression signatures with single cell data, but I think that there's a whole rich universe in the splicing data as well. That should be very exciting. Um, okay, so yeah, just to close out, I'll just mention that kind of bigger picture, up to now, Illumina has been mostly known for its sequencing instruments, but increasingly, I think we can also provide really nice software solutions and scientific solutions for interpretation. So I think most recently, all of us, Vanderbilt, UK Biobank, and Broad have all sort of moved onto the Dragon platform. So I think that's really exciting because it means that all these large data sets can now be analyzed uniformly across all of them, since it's a bit of a nightmare if someone's using, Dragon, someone's using GATK, someone's using something else. So that should really help for these kinds of large scale analyses to make them compatible. Yeah, and I'll finally just conclude by saying that these algorithms, they're all publicly available for and free for academic use. So you can just follow those links to get access to the tables. And yeah, this is our most recent preprint on the experiments. So I'll close right there and take any questions.